start a little differently today because I did this with the last class, and I did this with the class I do on Wednesday. It's so hard to get a context about the complexity of the cell without really getting an idea, at least a visualization of what's going on. I mean, I spent the whole, if you're looking at things two dimensionally, it's hard to see just how much stuff is going on inside of a cell. And I mean, this animation has been around. Harvard put it out. I don't know when it was actually made, sometime perhaps prior to 2000, but I've been using it since roughly then, over the better part of 25 years. I'm sure we have a more advanced understanding at this point, and you can find it. Originally it was just by Harvard. Now everything is YouTubed. So this particular one, if you want to see it again, it's going to be incorporated in our recording. However, Harvard Cell Animation narrated, so you can take a look at it. You're not going to be responsible for integrins and glycosylation and all of the intricate terminologies that you're going to see here. But it gives you an idea, particularly toward the very end, of how we talked about it, how white blood cells basically make their way out of a blood vessel and to a site of inflammation in the tissues. It shows the interrelationship between tissue and bloodstream. It shows all the intricacies of things being assembled and disassembled. So we'll begin there. They don't stop until something changes. Everything continuously moving. See the shape. So it's highly organized, even though it moves so freely. That's like the undersurface. This is assembling and disassembling a film. Motor proteins change shape. Vesicles are just being carried. This is going to be a free ribosome. It's coming right over there, that green guy. Here comes the bound. Watch how it slithers through. I love that. Exercise test. How it moves through the blood vessel. See, it's a the level of complexity, which I'm sure is better understood now, but it just gives you sort of a glimpse at just how complex it is. And I mean, and now you have something you can think of in your mind and go, there's a lot of stuff going on in there. So this is where we kind of left off. And effectively, the distinction, the endoplasmic reticulum is part of what's called the endomembrane system. It starts at the cell membrane, and it's a series of membrane tubes, membrane cul-de-sacs, roadways that protect stuff that's coming out of the nucleus and is going to be effectively somehow used in an area where it could be damaged by parts of the cell. You saw some of the stuff will go right into the cytosol or cytoplasm. That those messenger RNAs and proteins go and basically are picked up by what are called free ribosomes. all the ones that are either going to be, we talked about like a lysosome. There's going to be the proteins, the digestive proteins and enzymes in a lysosome. They're going to be in the endomembrane system. If they're going to be ones that are going to be part of the cell membrane or membranes of other of the membrane-bound organelles, they're in there to be protected. If we're going to spit them out, they have to be protected. You're going to see the three different ways that they're directed when we get to this Golgi apparatus, which is, again, sort of like the shipping department. It packages it, ships it, and makes sure is it going out FedEx, is it going out UPS, are we going to mail it? All of those types of things, that where things are going to be located. Nevertheless, that's the start of this endomembrane system, this remarkable system that protects things that need protection from the harsh interior of the cell. So you have, and the distinction between rough or smooth is a physical appearance, is a visible appearance on a microscope. Those bound ribosomes that I made mention of briefly before attach to those little pore areas, those little docking sites in the endoplasmic reticulum. They're always first. The smooth endoplasmic reticulum is always second. So when you see an abundance of these, and there's both in all cells, when you see an abundance of the rough, these are typically making proteins and polypeptides for export. Think of a pancreas cell making insulin. It's just filled with a vast amount of rough and a little bit of smooth, because you have to have something that's going to be able to make more membrane and process more membrane. Okay. If you go to a liver cell, as you will see shortly, liver cells have a modest amount of rough. They have a ton of smooth, because in smooth is where we process fats. That's what the liver does. It's where we detoxify things, which are mostly done in a way that, because they're so compatible with fats, it's easy for them to get in and out of our cells. That's what the liver does. It detoxifies. Okay, so and that's really the tragedy when somebody poisons their liver, frequently by alcohol, but by other things. And also it makes a variety of these neat little cul-de-sac things. We call them cisterns. A cistern is the old term for a well. And these little cisterns are really like a cul-de-sac. And you'll see that they play a pivotal role in calcium storage, in skeletal and cardiac muscles. You're going to see a lot of these. And that's what we're trying to build, this idea of where these things come from and what their uses are. So we move on. The rough, it appears rough. It's visible. Proteins that will be secreted from the cell, plasma membranes also, some phospholipids. They enter, wind their way, and these are a tubular system of membrane filled with fluid, and finally they're enclosed in what's called a vesicle. And you saw those motor proteins. It's not like magically they leave the endoplasmic reticulum. They just pick up some membrane coating and are trafficked by those motor proteins based on a variety of chemical signals to the appropriate area for processing. And once they're processed, they go back on this roadway, and this roadway is made up, as you will see, of more microtubule channels. It's a very busy area. So the smooth, continuous with the rough, just doesn't have the bound ribosomes. It makes enzymes found in plasma membranes, various things, lipid metabolism, cholesterol, making lipoproteins. That's, again, poster child for this is surely the liver. Absorption, synthesis, and transport of fats, the liver. And if you're getting anything out of this little talk, it's to underscore the importance of our liver. And there's so many ways we damage it. And again, and I'm not being preachy about it for someone who drinks very, very infrequently. But people have to be responsible with regard to that. Plus, hepatitis is more common than you think. And hepatitis is in one disease. Okay, hepatitis means inflammation of the liver. Yes, mostly from alcohol. It can be from other types of toxins. And it's not one virus. There are many viruses that cause it. The ones we hear the most about are A, B, and C. They're by letter. Okay, and they're not the same. They're all different. And they're spread differently. And they have different degrees of severity and treatability. Again, most of that stuff you unfortunately hear about in micro, particularly if you're stuck with me. And I use the term stuck advisedly. Not a smiler in the bunch. I know it's the weekend, and I am very thankful that you guys in the morning classes, you all show up here. I am very proud of that. I mean, I have a lot of affection for this class and the class before you. Because why you would sit in a classroom on a Friday getting ready for the weekend and listen to some stuffed shirt, a stuffed jersey? Don't understand. Detoxification, all the glycogen we need for our emergency supply of sugars, a lot of it that goes to the body comes from the liver. We constantly have to have it because every time there's an emergency situation, and we'll get to in what's called the autonomic nervous system, this is where it comes from, and here are those holders for calcium. And you won't have to know all the ends. They're called cis and trans ends. We use that terminology now, interestingly enough, in a societal standpoint, as you hear commonly these days. But the Golgi apparatus has one end called the cis end. That's the one that's coming from the endoplasmic reticulum, and the trans or transporting end is taking it where it's supposed to go. So all it is more stack. You can see the illustration, and you're going to see the micrograph, just like it looked like in that animation. A series of these saccules, all made out of membrane, and everything it's doing is processing. And so it's transported. I don't care about cis and trans ends and traffic directors and blah, 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 like that. Anything that makes it seem to you, I just think packaging and shipping and taking it where it's supposed to go. Things like that. So here's what it looks like. It's consistent in every cell. This is part of all our eukaryotic cells. You can see this really just sort of sandwiching of membrane and things being constantly utilized, and depending, as you will see shortly, is it going to be excreted? Is it going to be spit out of the cell? Is it going to go in the cell's membrane? Is it going to the membrane of an organelle like a mitochondria? Or is it going to be just contained in a vesicle like a lysosome? And that's part of it. The part I'm glad I looked over there. Down the road, I don't want to confuse you why this is here. It's not something you're going to get tested on. Ribosomes, just as an aside, I said to the other group, our ribosomes, based on their density, it's a fancy term called Svedberg units. Scientists always like to name things after themselves. It's vanity. So we call them S units because nobody wants to say Svedberg. Our ribosomes are 80S. Interestingly, mitochondria, why the bacterial origin of those organelles and a circular DNA, they are 70S. And it's a really important distinction down the road when you get to micro. So, and just to understand that all ribosomes are not created equal. The S just has to do with their density. It's the foundation, one of the two major foundations of the domain system. When I got my doctorate, the research hadn't been published yet. I got my doctorate in 77. I know. And this research was published in 78. We just had kingdoms, animals and plants. The whole domain thing, eukarya and prokarya and bacteria, it didn't exist. And it was all from molecular biology. This is part of it. It framed it. Nevertheless, here's what it does. They can be what we learned about before, commented on with those V snares and T snares. Secretory vesicles, spit it out. Vesicles, transmembrane proteins, so it'll just fuse with whatever membrane it's going to be and contribute to that, or enzymes in lysosomes, and something you're going to see right away called a peroxisome. This shows you, gives you an idea. You can read through this. It shows you being effectively coming from the rough and smooth into the Golgi, very much like the illustrations you saw micrographically. So whether phagosomes and lysosomes, basically coming together, whether it's going to be independent in some other way, whether it's going to be exocytosed, membrane protein, whatever you want to do with it. The peroxisome. And that's where all that stuff comes up on the board. So it's all about taking powerful detoxifying substances that neutralize toxins. Lysosomes are all about digestion and defense. They digest or break down things that have been ingested. Everything that's been phagocytized comes in, phagosome, fuses one of those lysosomes, We talked about it before, either digested if it's a nutrient and utilized, or digested if it's a bad guy and spat out. Okay, all good. The thing is, and it goes back to the whole thing with the liver, these are the things that the focus point of modern cancer theory, modern aging theory, a lot of modern disease theory involves things called free radicals, unpaired electrons, and and molecules that have an abundance of these. Effectively, what they do is they slam into stable molecules and destabilize them. So whether it's something that's involved in mitosis, it can destabilize that process and effectively lead to potentially for cancer cells. It can destabilize DNA strands and potentially lead to mutations. It can destabilize DNA strands and cause them to not function properly. And that's something that we attribute, at least in part, to the aging process. And we haven't even finished it. You get them environmentally. You can't avoid them. Any oxygen utilizing organism, us, by definition, what we call aerobic, we have to breathe air with oxygen in order to survive, has to have a method of dealing with oxygen, because we always, and I'll point out shortly, create these toxic derivatives of oxygen. It's not intent, it's just part of the process. We have adequate capabilities of doing it. When is it a problem? When we bring things into our bodies that make excessive amounts of them. Alcohol to excess. The carcinogens and the toxins associated with smoking in excess. Okay, almost any of that. Anytime various drugs and things like that that people do electively, and even some things that are therapeutic in nature, you're putting a burden on these peroxisomes to be able to detoxify what are called toxic derivatives of oxygen. Can't help it. We always make them. And so, and that's really what, if you're looking at something that's sort of a buzz term today, and it's been around for a long time, it's just getting at certain points of empty. It's healthy lifestyles, all the stuff. You're going to all learn about that. Most of you who are going into any kind of a clinical field. People have gotten a lot less healthy and have to get more healthy. And it has to do with the things, consumption, utilization, and being less active. Basically, our computers do a whole lot of stuff. Nevertheless, I put this on the board before. A major metabolic activity, the way basically we generate energy. Take carbohydrate, hydrate C6H12O6. We have to have oxygen as the entry into the mitochondria so we can generate ATP. So we need the oxygen-containing molecule if we are going to function. If we don't have it, we're not functioning. And in the process of doing that, we generate carbon dioxide, water, and ATP. How does it work? All those carbon to carbon and hydrogen bonds. And carbs have a bunch of them. Fats are even better because they don't have a whole lot of OH bonds that you really can't utilize in your research. Okay. They're filled. They're nonpolar covalent. They have a ton of energy to bond them together. Okay. And so when you peel them apart, you release a ton of energy from those pair of electrons. What you're left with are electrons. What are you going to do with those electrons flying around? You got them a parking place. That's right. Oxygen. Terminal electron acceptor. So remember oxygen, periodic table sitting over there. Oxygen has a valence of 2. Means it's not that it wants to lose 2 electrons. It's got 6 out of 8. It wants 2 electrons. There's oxygen. Take a half of an oxygen, go with it, and a couple of these unpaired electrons of the trillions of molecules of oxygen that are constantly being utilized, and effectively the abundance of electrons that are there that have had the energy sucked out of, they got to have a place to reside. Oxygen saying there, I need 2. So it's effectively bombarded. You would be naive to think that every oxygen would get the 2 electrons it needs. Because of that, we produce a fair amount of these guys. O2 with a minus sign, superoxide radical. That is the poster child for bad free radicals. That's the worst one of the bunch. That will go in unstabilized things. Again, it's what we worry about. We talk about basically initiators and promoters. Initiators cause damage. Promoters make the damage continually worse or worse again. And all the things I talked about, oxygen, carcinogens, they're all both. So don't be a doofus like me and have done bad things to yourself over time. Anybody here who smokes for 40 years and 2 packs a day, I want to know about it. See, none of you. I thank you. Sorry. So I'm here to give you this lecture for one reason. I have the capability of doing this for one reason, and it's my wife. I promised to her, before even when we were just engaged, before we were married, that I would stop smoking when she got out of nursing school. And I did. Moving on. So, peroxisomes, free radicals, highly reactive natural byproducts have to be detoxified. Havoc is not even a strong enough term. And this one is called, it's got a fancy name you'll learn about down the road called superoxide dismutase. And what it does is it changes it to something called hydrogen peroxide, which all of you have heard about. And most of you have it at home. Hydrogen peroxide is a great anti-infective. What happens is anytime tissue is disrupted, if you put it on intact skin, nothing happens. If you have a little scrape, you put it on, it bubbles. It also stings. What happens is anytime you disrupt cells, you release those contents. The contents of those cells will always have something called catalase because it's part of a peroxisome. They're disrupted. The catalase rapidly changes peroxide, hydrogen peroxide, to water and oxygen gas. That's the bubbles. It does so, and why it's so effective at killing bacteria. Bacteria don't like to be, they don't have the capability of dealing like we do with a ton of oxygen, rarely. That's how they work. Why does it sting? It's a very rapid reaction. It's very, very strong, and people don't realize the best way to use it would be to dilute it about 1 to 10. You could just take regular filtered water out of like a Brita or something like that, even tap water for that matter. 10 ounces of that, 1 ounce of hydrogen peroxide would be just as effective as out of the bottle is really too strong. Yeah, I talk more about that in micro when we start talking about how the utilization of it is. And yeah, they play a role in fatty acids. So I really think it's a vital point to understand. Here are the lysosomes we already addressed. They call them a safe site. It's safe because it's encased in membrane. Okay, and it isolates it from the rest of the cell if it's got those bad guys in it. So it does play an important role. Organelles do not last forever. I mentioned mitochondria can replicate it. When we stop replicating them, they have a lifespan. They're going to be degraded. That's where lysosomes come from and other organelles that are there. They play an important role when we have to release glycogen as an emergency sugar supply. And very, very importantly, and you're going to get to that soon, because our next unit, we jump right from the cells onto bone. My guess is we'll start that the week after this upcoming week. So we'll probably start that in and around whatever date that turns out to be, the 6th or the 8th. And so you're going to see that plays a really, and when you hear about apoptosis, programmed cell death, cells that are starting to go bad, we destroy and we basically recycle the elements. It's called autolysis. Cells in mitosis and meiosis that have major defects in them, and we don't want them to create a cell line. That's part of the way we are able to fight cancer and to fight disease, undergo autolysis. That's how you pronounce it. So if you want to say it, autolysis, okay with me. Lysis means splitting. The term that you're going to have to recall for a test is called apoptosis, APOPTOSIS, programmed cell death. It's kind of like why the tadpole loses its tail. Yeah. And I mentioned this disease before in the process of the importance about how lysosomes, it's not enough to just destroy stuff, they have to get rid of the garbage. This is effectively the classic example. And this happens in a particular missing an enzyme, and involves nerve cells in a developing brain. These glycolipids that they can't get rid of from the breakdown during just normal activities result in this defect. And again, ethnically speaking, my first wife and I were tested. We did not carry the trait, so it wouldn't have impacted us. Thankfully, if we were able to have children biologically, which we were not, but that was certainly one of the factors that could have been a deciding factor. And unfortunately, again, first 12 to 18 months, beautiful, happy child, and all of a sudden deteriorates and dies. It's just absolutely. And I have not known anybody. There's just so many anecdotal stories about this. And then the endomembrane system, you saw that passageway, all those motor proteins lugging giant vesicles. You saw the difference in size. That's very realistic. All I can think of is like ants carrying massive amounts of stuff. They can carry tremendous amounts, typically in my kitchen. Garbage disposal broke yesterday. I was making effectively a noodle pudding. My wife's church group last night. Hope it worked out. It was a great recipe. And all of a sudden, I'm trying to down the garbage disposal, looked under there. All I see is wet stuff and wires. Wires and wet is a very bad combination. All I can think of is ding, ding, ding. Here comes the plumber. So all the stuff that they do, the membranes all work together, blah, 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 blah. I said, to me, even though we see this slide all the time, it just doesn't tell the story. I don't know if this is terribly useful. Listen.
Protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein.
That was it. Keep going.
The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
Yeah, that's about it. It's not a whole lot to say. You saw it. That's why the animation first. There's a tremendous amount of structure. Some of it is always there. Some of it forms and unforms. That's because a lot of the elements have the ability to break down, move, and be rebuilt somewhere else. And so you're going to see that microfilaments, which are the smallest by diameter, microtubules, which are the largest by diameter, their building blocks, when they're not all coming together as a long polymer, are soluble. When you start to chain them together, they become insoluble. The one in the middle, diameter-wise, is also the one that's always there. The major structural agent is called an intermediate filament. The intermediate filaments are a tetramer, meaning it's 4 of those strands of protein that are braided together. Where do you see those? A lot of places. One thing that you have to know about with regard to those are desmosomes. The tetramers are really the basis of the fiber that skin is made out of, called keratin. And indeed, keratin filaments help to reinforce those desmosomes, those plaques and rivets that hold cells together very, very strongly. So that's why these intermediate filaments are there. The inside of the cell is an organized area. The parts of it are always ability to subject to change. Even things that are tethered, held, they're loosely so they can still have movement, as you saw with the mitochondria. It gives you, and the watchword for all of this is dynamic. Every time you hear a presentation, see an animation, the word dynamic, always moving around. So the filaments are made of actin, a protein you're going to hear a ton about because it's the major protein, one of the major proteins rather, that make up skeletal muscles and also smooth muscles and cardiac muscles. Actin, in its globular form, these individual units, is soluble. And when it's assembled, it's insoluble, it becomes fibrous. We even represent it that way. And you saw those things being assembled and disassembled that are there. Semi-flexible. The part that's always there is sort of the underpinnings of the outer membrane. Okay, but when you can build on that, it makes those tentacle-like projections that push the cell out, like for amoeboid motion, for pseudopods, for things that you'll see shortly called microvilli, which really just increase the degree of surface area, particularly for digestive cells. Very unique arrangement, gives a certain identity to a lot of cells. That's dense and cross-linked on the interior or cytoplasmic side. So they're a strengthening agent. But a lot of the others are for the cell moving, amoeboid motion, shape changes, pseudopods, endo and exocytosis. I always use to me, it's a double string of pearls. When it's like this, it's insoluble. You can see it, tangible. When it's just these little balls, they dissolve. The intermediate, rope-like, tough, strong, desmosomes. Nerve cells have them. That's the way they can conduct neurotransmitters in one direction. They can enclose other materials in another direction. There's a really intricate roadway in nerve cells. It's not just like an electrical wire. It's just, again, understanding that we use analogies for things, but they're not necessarily analogous structures. An electrical wire will send the impulse to wherever it's going, but in a cell, there's an internal composition that facilitates that. And here's what it looks like. Even though it's got more than 4, it's still a tetramer. I'll buy that. Maybe they mean there's 4 inside here, probably do. And again, that's in between diameter, and here's the only one that's somewhat hollow, and the only one somewhat hollow, and the one that has basically a different chemical, and they're the largest diameter. It's called tubulin, not overly creative naming. That's there. That's the protein subunit. Same deal. When it's disassembled, it dissolves. When it's assembled, it forms, and it's insoluble. They all come from that area near the nucleus called the centrosome. It's got all those microtubules that make up the spindle. All those microtubules that you're going to see make up cilia and flagella in our cells. Cilia in all of our cells, flagella in the sperm cell only, are all significant. Yes, some organelles that keeps them tethered. That the motor proteins really, the tracks are made of these microtubules, and depending on where we're sending it, you may have to build tracks. And if you build tracks in one place, you have to disassemble them from something. So you can see the way it is. Inherently, it's this sort of spiral. So there's hollow in there. The measurement is comparatively large by nanometers in diameter. Okay, and the motor proteins just change shape, powered by ATP. The centrosome is where it's located centrally near the nucleus. Centrioles, and they told them whatever shape you want to give them. I don't think the shape of the centriole, but that's where the microtubules come from. And what you're going to see when we talk about these are called intracellular appendages. It's not like a hair that protrudes. All of these, wherever they're made, those microtubules, when they're pushed out toward the edge of the cell, basically they take membrane along with them. Intracellular means if I stick it in here, I have a protrusion. In this case, it's encased in this shirt. Okay, but effectively it's got a sheath of membrane. So it may look like a projection, like a hair. The reality, it's still inside the cell. And that's a really important distinction. We don't have those. When you hear about flagella and bacteria that make bacteria move around, they move differently and they truly protrude like hairs. Because that's the way they have to get around for the most part. Classically, the centrioles, and this is a really important basis for, how shall I say it? This is a really important basis for what we would call evolutionary biology. When you were in high school and looked at things in pond water that moved around, that had flagella and were moving around, or amoebas, when they looked at the flagella or the cilia of those organisms, those tiny single-celled organisms, they had this arrangement. It's called 9+0. It has 9 triplets of microtubules with nothing in the center. This is at the centriole level, or the base body that attaches the flagella or the cilia in those organisms. The cilia and flagella, and not so the microvilli, which have intermediate filaments. The cilia effectively, and the cilia, they don't spin. They're just moving back and forth. It's a whip-like motion for cilia and flagella. In cilia, they're moving mucus principally, most of the time in our respiratory tract, trapping things so we can expel it. Or flagella, which are longer by definition. And again, the tail of the sperm isn't like a propeller. It's moving whip-like like that. They're made up of microtubules. The basal bodies have that 9+0, but inside it has a very famous 9+2. So when we have analyzed and been able to look at the tails of those lower order organisms, they have the same 9+2 thing. That 9+2 is really something that's very consistent evolutionary. If something in pond water has it and we have it, it really tells you a lot about justification for evolution. Same thing with sequencing of DNA. So there's a lot of science behind it. And I've said, maybe I said to you before, where Darwin came up with a sound theory. Unfortunately, the examples he used from the Galapagos weren't great examples. They suited him, but scientists have poked holes in those examples over the time, but not the theory when it's been examined, particularly molecular biologically. And I'm not an evolutionary biologist. I'm not the one qualified to talk to you about that. Here's good old 9+2. Here's 9+0, 3 triplets, 0 in the middle. Here's 9+2. 9+2 is coming your way in a test, not this time, but next time, I suspect. Got it? People are starting to finally come up to me and go, how do we study? What's on the test? I'm telling you what's on the test. Here's a whip-like motion. Here's the mucus. Don't get anything out of that. Microvilli, digestive cells, surface area. Those brush border enzymes. That's what it looks like. So these are made out of microfilaments. They're by definition thinner. They're more numerous. This is a very good illustration to show you effectively surface area. Your gut has all of these adaptations to increase surface area. And here you can see that web that's sort of like the underpinnings giving it structure, and they're all in these and just pushing out cytoplasm, and this is membrane, and effectively it just gives more room, more space for things to happen. And we're not going to do the nucleus today. We'll end it now. I want to end the same place with all the classes. What we'll end up doing on Monday is the nucleus, and we'll get a head start on the cell cycle, and we'll finish up that material after the exam. Thank you very much. Have a great weekend.